Hello, friends. Welcome to Developmental Biology. Vanessa Gordon says hello to everyone. She's sweet. I like Vanessa. Did I say it out loud? "22 people. So today we're just having a review. Let's see all your faces while you're here, just so we can pretend we're a class for a moment. Alex McKieran, I thought you were a guy for the longest time. It's like when all I see is the name, I don't know. I mean, I got a Sydney in the class and it's a girl too. It's just, Jesus. It's hard to be a teacher anymore. Jordan, for God's sake, I hope Jordan's a guy. He does too. What do we got here? 25, that's pretty good. We even got Kwame to show up this time. Yeah, Kwame. That's my friend. At least I think so. Okay, so this is all just about review. Do you guys have any questions you want me to answer? Our exam is going to be similar to last year's exams. Probably 60% of it will be kind of objective, you know, fill in the blanks and drag and drop type questions. Hang on one second. So, and then there'll be 4 essay questions, and the essay questions are the more complicated ones. They're less, you know, spitting out what you memorized and more like working with the problems and thinking about the answers. Let me take a quick look here on Moodle. I forget now what I have in your Moodle page for. Oh, I do have an exam there. Chapter 4, lecture 1, wait. Practice exam 1. So we have a practice exam there. What else is there? I guess that's it. Let's see what this practice exam looks like. So the practice exam is the one that I gave in class last year. There's a bunch of fill in the blanks and then some essay questions. So is the test Friday? Yes, it is. Oh, I forgot to tell you the results of the thing. Yeah, test is Friday. So, yeah, you did. Did I? Okay.
You did.
I don't know what the hell's going on. I just wish somebody was in charge. That's all. Oh, wait, I'm in charge. Shit. Okay. I mean, you did not. Oh, I did. I did forget, is what you're saying. Yeah, I forgot. Yeah, so the class will be Friday. Our test will be Friday. So do you guys have any specific questions you want? I can kind of go through the PowerPoints again, or we can go over that, the practice test. Anybody have any input here?
What time on Friday? Is it during class time?
Right, same time as class, 1 o'clock. And I think I made it an hour and 15 minutes long. So. Is it open book? Yep, there's not much I can do about that. It's online, so it's open book. So what I always say with open book is, you know, it's important to know where the information is. You know, you don't necessarily have to memorize it all, but know where it is and go to the book really quickly and refresh your mind, you know. Understand it, you know, understand the concepts and how things are moving and changing everything in the embryo. And then if you need to be reminded of some of the vocabulary, you can go back real quick and check it out. And you don't forget you've got that chapter 3 vocabulary thing. I've got a sheet there that's all the vocabulary from chapter 3. Make sure you go through that and understand those. You don't have to log into Zoom. It will be just just you guys alone, hopefully alone. What is the difference between neurulation, xenopus, and zebrafish? So the big difference there is xenopus is just like the chicken, the human, and the mouse, where you have neural plate, and neural plate rounds up into neural folds and it forms a neural tube. The difference with zebrafish is it kind of, instead of rolling up and forming a tube, it kind of closes from the bottom, so there's no hole in the middle, and it forms this neural keel, which is kind of like a V like this, but it closes up the V part of it. And then once it's closed up, then it pumps water in the middle and it forms a neural tube.
And that's the answer you're looking for too,
Yeah.
for a question like that?
Right. Can I go over convergence and extension? Sure. That is chapter 3. Oh, why do I have chapter 4 open? I don't need 4. Chapter 3, I'm going to converge and extend. Oh, let's do this real quick. You know, the first time I was doing Zoom, I'm talking on Zoom like this, and then I pull up my PowerPoint. I started talking about the PowerPoint, and someone chatted and said, "Dr. Slish, we like looking at your face, but we'd rather see your PowerPoint." It's a learning process. Come on, you. Okay, here we are. Okay, so here's that zebrafish question. So you've got this stage in the middle called the neural keel, where you've got no hole in the middle, and then it closes up with no hole, and then it fills in with fluid. Okay, convergence and extension. Oh, I'm into avian already. Shield region, involution. Okay, so the idea with, I don't know if you can see it here. Here, this is a good one. So similar to xenopus, it's a ball. You know, the chick is different, the mouse is different, but it's a ball, and you've got like that dorsal lip. In xenopus, we call this a dorsal lip. Here, it's called the shield region. So at that shield region, cells are dividing on the top. The ectodermal cells are dividing and they're coming down. They're hitting that shield region and they're going in. So they're like converging on that spot, and then they extend anteriorly and laterally. Okay. So there's your shield region. Cells are dividing, you know, ectodermal cells are dividing, and then they converge here in the middle at that shield region, and then they extend the shield region. And the extension is kind of how the embryo gets long like a fish instead of round like a ball. So that extension comes up this way. And it's also going out, doing the outer body cavity of the animal. Okay, so convergence means all these cells are as they're dividing, they're coming together at this spot over this way. And then they go through and extending up across the top, up across underneath the ectoderm, and then out sideways. Okay. Convergence and extension. Okay. Does that make sense, Marie? You got that? Good? Okay. So let's go, as long as nobody's got any questions. If you have any questions, please just chat and stop me. But as long as nobody has questions, let's just go over this PowerPoint from the beginning and stop me anytime. So the very beginning is just general stuff about body axes and how all chordates have got the same kind of basic setup, or all vertebrates anyway. Got a backbone, this axial skeleton or shoulder skeleton, and the pelvic skeleton, and then your appendages coming off that. There's the axial skeleton, is the middle part, the whole middle. Appendicular for the appendages. Radial symmetry, so the egg has radial symmetry, the You have radial symmetry, especially you see that in Xenopus at the early zygote stage. Chat. Chat. Give me my chat. Damn it. I can't see the chat when I'm sharing my screen. There it is. Oh shit, it just came up. What is a general term for multinucleated tissue? That's a good question. So that's called a syncytium. SYNCITIUM or something like that. Syncytium. So we see that quite a bit. I'll point that out as we go through syncytia. That's plural of syncytium. See the way I did that? A little bit of Latin. Okay, so you got your three axes, anterior, posterior, dorsal, ventral, and then left and right. So this is kind of nice. This shows you the different shapes of the early embryos in our three different, or well, five different things. Four model organisms on the human. So your Xenopus has got a big blastule with a big hole in the middle here, that blastocele. You don't have the blastocele in the zebrafish. And then with chick, it's just a disc at the top of the yolk. With the human, it's again a disc at the top with a big blastocele in it. The mouse, though, for some reason, this part comes down. So you end up with the blastocele in the center, and it's kind of a cylindrical shape. So that's, remember that about the different shapes of that early embryo, that blastula stage. I don't know why, but I can't see the chat. How about Sam? Can you read that last chat question?
What's the term for the movement of individual cells to the primitive streak?
That's ingression. When in the Xenopus, it's involution because it goes as a sheet of cells. In the chick, it's individual cells, and it's called ingression. Okay. Okay, so where are we? This is, again, talking generally about neurulation and how right here you've got your notochord. Remember, the notochord is mesoderm, and that's going to help to induce that ectoderm into neural ectoderm. So at this early stage of neurulation, you've got neural ectoderm here that the notochord is induced, and then you've got epidermal ectoderm on the outside. And this will form these neural folds. Neural plate will form neural folds. Somites will form at either side of it. And then the neural folds close up to give you the neural tube. And then you've got a somite, notochord, somite. Later in the class, we'll see how the somites turn into other body, the body wall and everything. Oh shit, I hate when I do that. Okay. Xenopus, I like the Xenopus. It's not the best model, I guess, for human because it's pretty different, but it's pretty clear. It's easy to work with, and when we get into the hormones and stuff, it's pretty obvious what's happening here. So important thing to remember about this is your first cleavage, you get two identical cells. Second cleavage, you've got four cells, but they're no longer identical, and that makes them pluripotent. These are totipotent. And then your third cleavage goes around this way, and that's the first time you get ectoderm versus endoderm. And then right on this margin here between the endoderm and the ectoderm, you get some of the ectoderm turning into mesoderm. And we'll talk about that a lot in the next chapter. So you end up with mesoderm around the middle. And now you've got your three different tissues: ectoderm, which is skin and nerve; mesoderm. I taught in the Kaplan MCAT course once, and they called it mesoderm, kind of as a mnemonic. because it's a way, it's a means of getting around. Everything that helps you get around is mesoderm. So mesoderm is bone and muscle and blood. Keep that in mind. And then endoderm is just gut. So this blastula then at one side, like we talked about on Monday, your sperm came in over here. So over on this side, we're going to form that dorsal lip. And the dorsal lip will be the organizer that's going to change cell type from different cells that go through. So here's your involution of cells. So basically ectodermal cells are coming in and turning into mesodermal cells, and they're dragging endodermal cells with them across the roof of that blastula. And that's giving you your three layers: ectoderm, mesoderm, and endoderm. And then this gap in the middle between two layers of endoderm is the archenteron, which is the primitive gut. That becomes the gut. So basically, this mesoderm is bringing some endoderm with it. The rest of this endoderm was there already over here. So that drags that through, and now you've got, endoderm on either side of the opening of the gut, which is what you want. At the same time, you've got a pibli going on, so this ectoderm is dividing and coming from either direction and covering up the whole embryo. So by the time you get to this stage, you've just got a hole in the bottom, which is going to be the anus here. You've got this blasto plug, yolk plug, and the blastocele is going to get pushed out that way and leave. Okay, so that's gastrulation. Oh, here it is again. Blastocele, marginal zone, dorsal lip, archenteron, yolk plug. Okay, and then neurulation happens just like we saw before within the general scheme, where you've got neural folds, first neural plate, which then becomes neural folds, and then close up for neural tube. Now you've got somites on either side. Now, where is it? Okay, there it is again. Oh, another important thing here in this neurulation is when that neural tube closes up, there's still some neural or originally neural cells at the top of it between the ectoderm and the neural tube, and those are your neural crest cells. And those are important because they're going to migrate away. That's another good vocabulary term, mesenchymal. They're mesenchymal cells. And that means they're loosely organized. They're not a sheet of tissue. They're individual cells. And they crawl away separately and they make different things. So they will be peripheral nerves. They're going to be melanocytes, which is really freaky to me. Because now they're going to go out and go into from that one little spot, they go all over the body and they give your body color. So that's kind of an interesting migration of those things. And I often think that may be why, I don't know, I'm making this up, but that may be why melanoma is so dangerous, because these cells are so migratory already. So if they become cancerous and they go back to their migratory stage, they metastasize really quickly. That's just my hypothesis, but I'm rarely wrong, as you know. Okay, so this is important here. So these are your, so there's your notochord, and you got the somite on either side. Now the somite starts to split after neurulation, and you end up with somatopleural mesoderm and splanchnopleural mesoderm. And each of these will become different things. I believe the somatopleural then is, oh boy, I hope you guys know this by Thursday, because I have just forgotten it. I can't believe I've forgotten this. Zebrafish, that's not what I want. I was going to say it. We'll tell you what, I'm going to say it and then I'll look it up and I'll tell you, and you'll see if I am always right or if I screwed this up. So this splanchnopleural is going to be the body wall, and the somatopleural would be like the appendages, the arms and stuff. That's what I'm thinking. Let's see now. Bum, ba, bum, 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 bum. Bum, ba, ba, bum, ba, ba, bum, bum. Neurulation. Okay, let's get a new share. I'm not even going to read it. We're going to read it together. Okay, somatopleural becomes body wall and limbs. Okay, so I put both body wall and limbs in one. So that's going to be body wall and limbs. And then this is going to be the ventral internal organs, heart, blood, mesoderm, and foregut. Okay. All right, so that's what those two different things go. And although I don't necessarily know it off the tip of my tongue, you better by Friday. That's called hypocrisy. Okay, now we're into zebrafish. Okay, I had another drawing of this. I think there's one in your book that I thought was better. It was like a electron micrograph of it. Hang on a second. There it is. Okay, so again, neural tube, somite, and then here's the rest of the mesoderm that's going to be somatopleural and splanchnoplural, and a coelom in between. So that coelom is like the space between stuff that still exists in your body, space between the body wall and internal organs. Okay, so let's go back to where we were. Go away. Okay. And then we get to tail bud stage, and we haven't really talked about tail bud stage. That's okay. I just want to get you through neurulation at this point. Maybe we'll talk more about this at the end of the class if I run out of notes and we need to talk about other things. Okay, so having done a frog, we're going to go back in evolution and do a fish. Probably should be in the other order. So the difference here, big difference is we still have a ball, but it's not a hollow ball of cells. Most of the cells are on top. And that is, so your embryo is up here. I guess what's interesting about this is now you can see a difference between cells that are going to be extra embryonic and cells that'll be embryonic. Here you've got your enveloping layer, which is extra embryonic. This yolk syncytial layer, there's that syncytium. So these are cells that fuse together, so they're multinucleated. Here you've got mesendodermal cells. So these can go either way, mesenderm or endoderm. And then here's your mesoderm and your ectoderm. And here is that epibole happening. The ectoderm is coming down on either side. And then here at the main organizer shield region, you're going to get that ingression. Is it ingression? I think it's involution here, involution around this germ ring. And it's happening, that's another difference here. In Xenopus, it only happens at the node over here. In your zebrafish, it's happening 360 degrees around, except it's mainly happening here. Most of it happens here. And this is where your convergence and extension happens. Okay, so cells are coming in, converging, and extending anteriorly to give you that stretched out body plan. Oh, and there's our neurulation again for zebrafish. I keep screwing up zebrafish and Xenopus. This is just comparing the two again. Okay, now we're doing a chick. So the big difference here with the chick is now this is very different from the other two because it's not just a ball. Now it's like this disc on top of the yolk. And this disc will develop a primitive streak. You all remember my really bad primitive streak joke. At least I hope you don't. Okay, so here we've got ingression again, where the primitive streak starts at the posterior end and extends up to the anterior end. And what it really is cells coming together and then ingressing. So that's what, and it makes a little dent as they're coming down and in, and that's what the primitive streak is actually the cells coming down and in. So as the primitive streak is extending, so the ectodermal cells that are dividing here are coming down and in. And remember, these cells over here are endoblast cells, and they are not going to be part of the embryo. They're part of the extra embryonic tissue. So every embryo of a vertebrate's got 3 layers of cells. It's starting out with just this top layer. When you ingress, then you get the other 2 layers coming with it. Where was the Okay, so why did I have this first? I don't know. I don't know why that's first, because we'll talk about that in a minute. Let's start here. Okay, so that disc here is on top of the yolk, and it's just sitting on top of the yolk there. Other extra embryonic parts of the chick are the yolk, of course, the vitelline membrane that goes around the whole thing and on top of the blastoderm as well. These things just stop the egg, the yolk and everything from swirling around. Egg white, the albumin. What's this membrane? Shell membrane, I guess. I don't know. I forget now. But anyway, here, this is kind of weird and different too. At the beginning part of your blastoderm, cells divide, but the cleavage furrow doesn't go all the way through. So it's kind of syncytial here too. Doesn't completely close up. So you don't have plasma membranes on the bottom of these cells, and they can directly interact with the yolk and get their energy from that. But then as it develops, those cells will close off and they'll form this subgerminal space. So this is the germ layer. This is what the embryo is actually. And you've got a space underneath it, your subgerminal space. And then these endoblast cells will come across it. So at this stage, you've got a dark area and a translucent area. So the opaque area is area opaca, and the translucent is area pellucida. And you can imagine if you were a scientist way back in the day, and all you can do is look at this, you would be like, oh, look, there's a translucent area and a dark area. Now we know what it actually is, and it's translucent because you've got an open space here. You've got 2 cells thick on top, and that's your germ layer. That's the epiblast. And then down underneath here, you've got your subgerminal space. And here we're forming those hypoblast cells. So the hypoblast cells are extra embryonic, and they are kind of interacting with the yolk. And then your epiblast cells then are the actual embryo that go across the top. And it's still thickened at the side, so you still have that that area opaca there. And then at one end, what turns out to be the posterior end, you get this Kohler sickle, which is thickening of these cells in the area opaca. And you want to say hello? This is my lovely wife, Erica.
Hi, everybody.
She's pretty. Anyone who doesn't think she's pretty, I'll take you outside. It's too cold outside. Okay. All right. So now Kohler's sickle then is where this primitive streak will start. So that's posterior, and it extends forward. And again, what that primitive streak is these ectodermal cells dividing and then coming down and ingressing in. And they're ingressing in as mesenchymal cells. They're not a solid sheet. They're individual cells going around. Some of them are going to be endoderm, some of them mesoderm. Okay, so they're kind of, and then as the primitive streak gets all the way to the end, when it gets to the end, Kohler's node starts here. Henson's node starts here. Okay, so primitive streak makes it kind of extend. So now you've got your long body axis, and you've got some endoderm and mesoderm giving you 3 layers. But now this is going to regress. So I always say it's like a zipper going up and then coming back down. So as it's coming back down, it's Henson's node. And whenever we say node, that means it's a bunch of tissue that's going to give off hormones and change cell fate of cells that go through it. So Henson's node is then going to regress. And as it does, it's laying down the notochord and the notochord is inducing the neural plate. So cells are coming down in through this node, and they're basically dividing really fast too out here. This kind of slightly darker area in front of the notochord is called the stem zone. You know, just like stem cells, dividing. So you get a lot of cells dividing here. They're going in through the node, and they're being specified as mesoderm and endoderm as they go through. And then when Hensen's node gets all the way to the other end, then you're done. And cells keep going through in the regressing primitive streak. That's your post-annual tail that happens after that. But something I think we talk about here is cells that are ingressing here are going forward. Cells are always going anterior and lateral. So these cells are going forward, and when they do, they start to form that head fold. I think we talk about that some more in a minute. And here you can see the somites forming. Somites are basically body segments, and we'll talk a lot about that later. Don't worry if you're not quite catching it yet. But those are just blocks of cells that will then go on to form body cavity and things like that. Body wall, I mean. Okay, so this is a drawing I made of that stem zone and Hensen's node. Cells are dividing here, and here they can be either ectoderm, they're kind of between ectoderm and mesoderm, and as they go through, some of them turn into neural cells and become the neural plate, and some of them become mesoderm and become the notochord and somites. We'll look at this in a lot of detail later, and we'll see the hormonal interactions that are causing this to happen, which is really cool. Okay, so this is like one embryo that's halfway through the whole process, and they cut it up. And then what I like about this is you can see the process by looking, you can see different points of the process. Instead of looking at one segment over time, you look at the whole thing in different segments, and you're looking at different points in time, because this is, this part is ahead of this part back here. So you can see here as it just starts, here's your neural folds, neural plate in the middle, forming neural folds. And the folds are coming up, and as they do, you've got your notochord and somites, and then here's that somatopleural and splanknopleural mesoderm. And now there's a cecum, and you're forming your body cavity here. This is almost closed up, almost closed, and it's never quite closed up here. And never forget your neural crest cells that came from here, and they crawl on out away from there. So that's your chick. Chick is pretty straightforward. The extra embryonic tissues here of the chick in mammalian, we'll see this as well, this amnion, which is the membrane that contains the embryo and the amniotic cavity. And outside of that is the chorion, which contains the whole embryo, including the yolk and some of these other structures. So here's the umbilicus, more or less. And you've got the allantois, which is going to come close to the shell, and close enough that you can allow gas exchange. And then the other part of the umbilicus is getting food down here at the yolk sac. Vitelline artery in red, vitelline vein in blue. Okay, so it looks like blood comes down this way. Some goes that way, some goes out here, comes back here, back into the heart, and back out. Okay. Comes the mouse. This cylindrical structure of the mouse is odd. I don't quite understand why it does that, but only rodents do that. Okay. 4-cell. Remember, no huge yolk sac here like in the other ones, because you're going to get a placenta. So 4-cell, 8-cell, and then after it gets to 8-cell, it compacts, so the cells are touching each other more, and then you go into this blastocyst. Morula blastocyst. Let's look at that blastocyst. So the blastocyst has got a lot of different types of cells here, and only some of them are embryo proper. The other ones are all extra embryonic. So trophectoderm are the cells that go on the outside of the whole thing. And the hole is always there. The fluid-filled space is always blastocele. So you've got mural trophectoderm down here and polar trophectoderm up here. This is the inner mass of cells. The blue ones are the epiblast. Epiblast, always remember epiblast is the actual embryonic tissue. Now here's primitive endoderm, which will not become part of the embryo in the end, but it's important at this stage. And then that starts to form this cylindrical type thing. And what's pushing down here is extraembryonic stuff, ectoplacental cone, the extraembryonic ectoderm. Okay, that's going to be part of the umbilicus. Here's the epiblast, that's the actual embryo, and it's surrounded by this visceral endoderm here. Again, extraembryonic. And on the outside, you've got parietal endoderm. So there's two more endoderms to memorize. Visceral endoderm here, and then parietal out here. And then the outside, this is really similar to what we see with the human trophoblast giant cells. So these are trophectoderm cells that fuse to each other, become like a syncytium, and they're big multinucleated cells. They're going to be the cells that help that embryo to implant in the uterine membrane. So remember that your trophoblast giant cells are what helps that embryo embed in the thing, in the uterus. Here we are a little bit later on, late blastula, epiblast cells here, visceral endoderm, parietal endoderm. How many damn endoderms does this embryo need? Well, it's going to get one more in a minute. So we're going to start up. Remember, this is going to be posterior. Oops, shit. This is posterior, and this is anterior over here. So we're going to start a primitive streak right here and have it come down, I believe. Oh, let's get rid of that naked guy. So this is like the node here. These turn into mesodermal cells, and they start to egress, you would say, instead of ingress, because it's coming outside. So it's coming out and going anterior and lateral, and it's going to cover the whole embryo in that way. And it's going to insert itself. It's coming out and going underneath that visceral endoderm, and some of it will be endoderm, and that will be I guess it's presumptive endoderm, like actual what's going to be endoderm, and that's displacing that visceral endoderm as it comes across. So cells are coming out through this primitive streak and going anterior and lateral. And that node will come all the way down to here. Some cells will continue to come through and go forward and become the head process. But this node is going to regress in a second. So here's your node. This is going to be brain over here, head fold. And here you can see your layers. Visceral endodermis on the outside. You've got other lost mesoderm is here. Okay. Here's your head fold, notochord, and the rest of the mesoderm there. Your primitive is green is the definitive endoderm, so that's what's actually going to be the endoderm in the end. Is there a definitive endoderm here? They didn't note it. It should be there though. Okay, and then boom, now you've got this flipped inside out guy with a definitive endoderm here in green, which is the actual endoderm. You still have visceral endoderm on the outside of this, but you can't see it here. They took it away. There's your notochord. And the neural fold, of course, is on the inside because we're inside out here. So the notochord is underneath with the ectoderm on the inside and forming somites here. Let's back up a tad. I don't know. Okay. And then, so it's all inside out. I don't know if these pictures help you, but this is the way I think of it. It's backwards, where the head is here, the back is bent backwards and with spine on the inside and the posterior end is over here. It needs to flip so that it's like this. So the head is over here and the tail is over there. And it does that through this turning process where first the head and tail come together and then some kind of magic happens, and I won't ask you about it on the test because I don't understand it, and boom, it flips. Ta-da. I think that's one of the kind of thing you would need to see in a slow motion video in order to really get, because I never really catch it like this. What the hell is this? But anyway, the head and tail come together and then it flips and then boom, and then you're good. So now at this point, this becomes your umbilical cord coming down into the endodermal gut here. And yeah, so your dorsal side is out here and your ventral's on the inside, which is what you want. Human development. Okay, so with human development, mainly what I talk about here is the development of the placenta, because it's not really discussed much in the mouse. And the actual embryo development is very similar to the chick. So it's not that much different. But anyway, let's start out here. You've got your extraembryonic mesoderm here. Here's your epiblast. And this will be the amnion that surrounds the embryo. Here's your yolk sac. You're going to start with a little bit of yolk. And hypoblast underneath the blast. So this is extraembryonic hypoblast. Extraembryonic mesoderm covering the whole thing. And that'll be the chorion in the end. Okay. So there's the yolk sac, hypoblasts here. And here's the blast cells. You get your primitive streak going up. Cells ingressing, forms a node, and here's neurulation, which again is very similar to everything but the mouse. You can see your somites and your splencto and splenecto plural. Okay, amniotic cavity where your amniotic fluid is. Here's the yolk sac, which is on the ventral side, right? And then this will become the umbilical cord that'll come into the ventral side where your belly button is. Okay. And here we can see the head and tail come forward. Yolk cavity, this yolk, you'll use up all this yolk and eventually this will form into the umbilical cord. Yolk's mostly gone. Blah, blah, blah. Okay, so here's what's most important for me about the human. Like here we're getting a better understanding of how that embryo is invading the uterine lining and attaching. Okay, so syncytio trophoblasts, you've heard of these before. These are the trophoblast cells that are fusing and forming a syncytium. And then you've also got cytotrophoblasts. So these are same origin, the cells are from the same origin, except they're staying as individual cells. That's why they're called cyto. These are fusing to make a syncytium, so they're syncytio. So your syncytios are going to invade into the uterine lining. So these pink cells are maternal uterine cells, and they're invading in. Once they invade in, they will start giving off HCG. And this is when you can detect that you're pregnant because you've got HCG being released. And this is a beautiful thing too, how like the embryo tells the mother, hey, I'm here. And the mother goes, okay. And so the HCG goes to her brain and causes a release of more progesterone. And that increases the vasculature in the uterine lining and helps to solidify that connection between the embryo and the mother. I think that's really cool, the communication back and forth between the two. Okay, so your syncytiotrophoblasts here are like one big globular cell that's invading. And then you've got your cytotrophoblasts that are still individual cells here. And then here is the epiblast here and the hypoblast underneath it. And then that will then develop into these villi. And the villi are these finger-like extensions into it. Again, you've got all these blood vessels on the inside of the villi and a lot of membrane space in that villi to increase surface area. Here's a close-up of it. The outside layer of that villus is cytotrophoblasts. Your syncytiotrophoblasts then are kind of this blue layer on the outside of that. And then this is that intravillus space. So space between the villi. And the mother's, this always freaks me out too. The mother's uterine artery just spills blood into that, that intravenous space or intravillus space. So these villi are just being bathed in blood. And so they can get a lot of great exchange of oxygen and carbon dioxide and nutrients and everything else because you've got only one thin layer here, the cytotrophoblast between the arteries and the intravillus space where all the blood is. And then you've got a constant flow of blood into this intravillus space and then back out through the uterine vein. I think this is a really nice view of it here. What I like about this view is here you can see oxygenated blood pouring into that intravillus space on the bottom. As it goes past the villi, the oxygen gets taken out of it, it turns blue, and then it comes back in this way and into the vein. So you're bathing those villi with blood. What time is it anyway? Uh-oh, class is just about over. All right, so there's that. And then the twinning stuff. I may ask you, why don't I just say now I'm not going to ask you a question about twinning. I think it's interesting, but we got a lot of other things to talk about. And this is good. You should definitely look over this, but I'm not going to stop now because we have 2 minutes left. And then you got techniques, which we reviewed on Monday a little bit, I think, or maybe we just gave you more. No, it was Friday we did techniques. So, I'm out of time here. I can't really talk about it much, but make sure you know these techniques too, because I will ask you questions about that. Okay. Let me just stop the share and see if we have any more questions. Okay, any more questions before we say goodbye? I'm losing people. Let's see, let's wait till Tyler Montano leaves and then I'll tell you a dirty joke. Just kidding, I don't know any dirty jokes. All right, see you later, guys. See you on Friday. Actually, I won't be here on Friday. You'll just do it yourself. All right, see you later.